Welcome. My name is Zhao. I am an assistant professor at the Johns Hopkins University Department of Biostatistics. It's my pleasure today to come here and share some of my work with a group of people who work on microbiology. I generally develop statistical methods for analyzing microbiome data. And today, I'm going to talk about a log linear model for inferences on bias in microbiome sequencing studies. This is joint work with me, myself, and Dr. Glenn Santon from Emory University. So I think it doesn't require me to say much about the importance about the microbiome in human health and in how it relates to our everyday life. So now many large studies routinely collect data on the bacterial communities in our body, such as gut and skin and oral. So generally speaking, the way to measure these microbiome abundances usually looks like this. We use sequencing method to do it, so that we can, instead of having to culture the different bacterial species, we can have the read count for thousands of bacterial species at the same time. The general workflow is like this. So we first isolate the DNA from the tissues, extract the DNA, and then those DNAs are subject to sequencing. Then there are generally two ways to sequence the bacterial DNAs, but those are not important. But after all, at the end of the day, what we get is a big table where, generally speaking, each of the rows represent a sample, and each of the columns represent one different bacterial species. Then we can use this table, which characterizes the microbiome community in the sample that you collect, to do many things, such as we may want to associate to see whether the microbiome community is related to human health. Maybe it's important for treatment, or maybe we can use it for prediction. So in the sequencing studies, bias is something that we cannot go around. Bias is ubiquitous. In microbiome sequencing, it is widespread in all types of high-dimensional genomic sequencing. Bias is defined as a systematic distortion of the measurement from the true values. So what you see from your sequencer is different from what the true relative abundances of the bacterial species in the community. Recently, there was this high-profile paper which characterized and tried to correct for the bias in metagenomic sequencing experiments. So generally, what they observe, we call this model as the MWC model, based on the three initials of the three authors. The general conclusion from this model, from their experiment, was that the bias is a multiplicative factor that works on the different bacterial relative abundances. So generally speaking, this is a toy example where there are only 3 bacterial species that are in the community. Suppose you have them, each of them constitute 1/3 of the system. Then during each of the steps in the experimental protocol, you do the DNA extraction, you do the TCR, you do the sequencing. Each of the steps has a multiplicative factor on the bias of the true relative abundances. And at the end of the day, what you observe is a distorted version of the relative abundances that are in the community. So what this multiplicative bias would mean to us with respect to analysis? One of the things that we can immediately realize was that because of the multiplicative bias, we may sometimes, the data that we receive may direct us to a different conclusion than what the truth it is. And this is true even when all the samples were processed in the same way, and all the samples have a same bias factor. So this is also a toy example from their MWC paper, that suppose you have 2 samples, sample 1 and sample 2, that have 3 bacterial species, that sample 3 has a reduced relative abundance in the true community. But because of the bias factor that are the same across the 2 studies, across the 2 experiments, you you see actually an increased relative abundance in the observed data, while in the true data, there's actually a reduction in the relative abundances of taxon 3. So this is problematic because generally speaking, most of the statistical methods that people use for analyzing microbiome data assumes that the bias can cancel out if all your samples were processed exactly in the same way. Well, it is not true based on the MWC model. So what we intended to do was the MWC model is very indicative, but there are still some problems with it. So what we want to do is we want to try to evaluate whether this MWC model works fine in our data set and to what extent this is correct. So the MWC model, if we write it in a mathematical formula, it generally looks like this, where there's a little bit of a notation change when I convert my PDF file to the PowerPoint. But generally speaking here, the P is the true relative abundance of a taxon J in the community, and P tilde is the observed relative abundance in the community. And the multiplicative factor generally says that, okay, my observed relative abundance is the true relative abundance multiplying some factor, where this exponential is just for mathematical convenience, but this is a multiplicative factor. Of course, you have to standardize it somehow, where the denominator is the summation of the numerator across all the J's, where the J is the total number of bacterial species in the community. So MWC model is a multiplicative model. It's also a deterministic model. That it says your P tilde is a multiplicative change of your P. Generally speaking, statisticians do not like deterministic models because we think there will always be randomness in the system, and we want to characterize this randomness to see how well the data with randomness will fit into this deterministic model. The second implication from the MWC model was that the bias factor of one taxon is not impacted by other taxons in the community. This is important because this allows us to model the bias one taxon at a time instead of having to think about the interactions between the whole communities. But there's also a question mark because the MWC paper did not really test on whether this assumption is true, whether another taxon may impact the bias of this one. The third observation from the MWC model was that the bias depends on both the experimental procedures as well as bacterial properties. So one taxon may have a low bias factor using one DNA extraction protocol, but it may have a high bias factor using a different extraction protocol. And reversely, in the same extraction protocol, one taxon may have a high bias factor, another taxon may have a low bias factor, depending on the characteristics of the taxon, which we think makes real sense because the taxon may have different cell characteristics, such as whether they are gram-negatives or gram-positives. And the final point was that the normalization factor depends on the true prevalence of the bias factors of all taxons in the sample, and we don't have any issue with that. The purpose of this study is really to try to say, okay, if we think about the random noise in the data, and we want to test whether there's real interactions between taxa and taxa with respect to bias. So the data set that we use is from the Brooks data, from Paul Brooks. This is a mock community with 7 taxons. This is a fairly large fairly large data set that have 240 samples with 58 combinations of bacteria. So essentially, they have 7 different bacteria that they mix with different proportions, where the proportions are known so that we know the true relative abundances, and therefore we will be able to model the biases. The samples were processed in 6 plates through 3 different ways, where we name it as cells, DNA, and a PCR product. This is where from which product the samples were processed. And the little table tells how many of the samples have only 1 taxa, how many samples have 2 taxa, and how many samples have all 7 of the taxons in the sample. So our research questions are like, do the plates have different bias between them? Do the different plates have a very different effect? And also we want to ask whether one taxon impacts the bias of another taxon. Of course, we know the data are presented in a way that there are many missings because that many of the species are not present in certain samples because of the design. So the model setup are generally like this. So we have an OTU table or an AESV table. Essentially, this is a species table where each of the row is a sample and each of the column is a species. There are N rows and J columns, where the P and the P tilde are the true and the observed relative abundances of taxon J in sample I. And we have an indicator delta Ij to indicate whether delta J is present in sample I. It is 1 if it is present and it's 0 otherwise. Of course, we assume that all the relative abundances in that sample sum up to 1, no matter in the true relative abundance sense or in the observed relative abundance sense. So this is just for taxonality. And we extended the MWC model. So you see everything on the right side is exactly the same as the original MWC model. The only difference was that we put P tilde, which is observed relative abundances, we replace it instead of the expected values so that we know, okay, there is randomness in this equation, and we only say on average, your observed relative abundances is that multiplicative effect on the true relative abundances. Okay, this extended model with noise can also be rewritten in something like in a log linear model, where essentially the log of the P tilde equals the log of the true relative abundances plus beta J, that is a bias indicator for each of the individual taxa. Then there's an effect for alpha I, which represents the samples, and this is from the common denominator from the previous model. And there's an epsilon where when I do the conversion, that epsilon disappears, but there's a plus epsilon ij. We further extend the model to allow for other characteristics, which are the xi's over here in the model to be characterized, such as the xi's generally represent in what protocol the samples were processed, such as in which batch or in which plate the samples were processed. So for estimation, this model is overly parameterized. There can be, so as later I will show using real examples, you will be able to see that we may be able to have multiple combinations of data that provide the same equally well fitted to the data that we have. They will have like the same different parameters that give you the same model fit. But for estimation, what we intended to do is that we intended to get one set of best estimators among the infinitely many parameter estimators. Well, we will be able to do hypothesis testing where when we properly set up the hypothesis testing, even though our data are not identifiable, we will still be able to test for those hypotheses, meaning that we will be able to ask many interesting questions. So what we observe is that if we multiply our equations by a compositional projection operator, which is essentially the diagonal matrix of the delta, which is the indicator about whether or not that taxa are present in the sample, minus 1 over Ni, where Ni is the number of taxa present in the sample, times the inner product of delta I, transpose and delta I. This will eliminate anything that is constant in I, and we no longer have to estimate alpha I. And the final model looks like something like this, where the Yij is a transformed version of the observed relative abundance, and the difference between the log observed relative abundance as the log true relative abundances times this compositional projection operators. And the E is the original epsilon times this compositional projection operator as well. What we do is that we use something like a vector trick. Essentially, the general idea is that our Yij is a single number, but if you look at it all together, your Y together is an n by T matrix. And all the operations is on the matrix form. What we do is that we stretch out all our Y's. So essentially, we stack the second columns of Y after the first columns of Y and the third columns of Y, and do this. And we can transform this into, we call this as a vector trick for estimation, so that what we estimate is a vector value of this beta matrix. And we can, once we get this vectorized of this beta matrix, we can convert it back to the original beta in the matrix form. There is some like a canonical product and some mathematical relationships that are needed, but those are not really important. The importance was that by using this vector trick, we will be able to run the estimation very simply using least squares. I can give some examples to really show why the model is over-parameterized. So suppose we have two plates. Our X will be two indicators. If the sample was processed in plate 1, it will be 1 in the first column, and if it's processed in plate 2, it will be 1 on the second column. Then our beta will be a 2 by J matrix, where J is the total number of species. In the Brooks data, that is 7. What we see is that if we add a constant to the rows, meaning that because the bias is multiplicative, if we add a constant, which means we multiply the relative abundances of all species in that sample by a constant, then the relative abundances do not change at all, because once you do the standardization, that it disappears. So this gives why the model that we have is an overly parameterized model. You add a constant, the model fit will be exactly the same. So another example is that suppose we are interested in testing the interactions, then the X matrix will have an intercept. Then you have a delta matrix, which essentially represents whether taxon 1 is present in sample 1 or taxon 2 is present in sample 1. And your beta matrix will look like this, where the first rows represent the main effect, and in the below, it represents interactions, where the diagonals on the below J by J matrix are zeros, because the main effect is already in the first row. We can also use an alternative way to specify this model, where we remove the intercepts of ones and only keep the indicators about whether or not that species is present. Then the diagonal element will represent the main effect. Again, we see if we add a constant in each of the rows, it doesn't change any of the bias. If we add a value to the diagonal element, it will not change the interaction at all, which also says that we have an over-parameterized model. So even though our model is overly parameterized, but we can do inference in the format like this, where we have C as a context matrix, where we have the vector of data, and we can test whether this one is 0. In most of the contexts, we can write this one as C beta Q, where the Q accounts for the non-identifiability in the beta matrix. So I'll give an example. So for example, suppose we have 3 taxa, no covariance, no missing data, then our beta will be, our beta matrix is just the beta 1, 1, beta 1, 2, and beta 1, 3. The vectorizer of it will be like this as well. Suppose we want to ask whether there's any bias in this system, which essentially asks whether the bias is different for taxon 1, taxon 2, and taxon 3. So your context matrix will look like this. So essentially, this -1 and a +1 says that, okay, beta 1, -1 times beta 1 plus 1 times beta 1, 2, so essentially equals 0. So that says beta 1, 1 minus beta 1, 2 is 0. The bottom is beta 1, 1 minus beta 1, 3 equals 0. When all three of them are the 0, which means there's no bias, because again, we know adding a constant doesn't change anything. And if the three of them are the same, we can think about them as all zeros. This is one example of that. Another example, suppose we have 2 plates as before. If we want to test whether the bias factors differ between the plates, which essentially says beta 1j minus beta 1j minus beta 1j prime, sorry, this is 1j prime, equals beta 2j minus beta 2j prime. So essentially the difference between the 2 rows in each of the columns are the same. This will be for the May effect model. And if we go on to even more complex models, such as when we want interactions, such as in the Brooks data, suppose we want to know whether taxon J impacts the bias factors of other taxa. That is to ask whether beta jk equals beta jk prime, where k is not equal to k prime, which is not equal to j. So suppose we want to know whether beta 1 impacts the bias factor of other taxa, which essentially talks about whether beta 1, 2 equals beta 1, 3 equals beta 1j. And for the Brooks data, this will be a 5 degrees of freedom test. And there are some other examples for this as well. If we want to test whether there's any interaction between the taxa, which is to test whether beta jk equals beta jk prime for all j, k, and k prime with 7 taxa in the Brooks data. This entails a 35 degrees of freedom test, which essentially says that we have 35 equations that we want to test for this system. So the inference that we do, we propose to do a F statistics, which is very simple to understand. So essentially what we do is that we can calculate the residuals under the null model, and we can also calculate the residuals from the full model. The full model is just the model that we previously presented. I presented the null model would be the model with a constraint that certain of the betas have to be the same, or certain functions of the betas has to be the same. So what we get is that we can get the residuals from this model, and we want to see whether the data, whether the models fit the data similarly. If the model fits the data similarly, then your residuals should be similar, and your residual sum of squares should also be similar. So essentially, the smaller the F statistics is, the smaller difference is between your null model and your full model, which also says that the full model is not any different from the null model, and we would prefer the null model because we can use we can put more structure into the data, and it models the data as good as when we allow for more flexibility. So the F statistics is just the residual sum of squares under the null model minus the residual sum of squares over the full model divided by the residual sum of squares over the full model, where it's just a function from all the residuals. The null distribution of this F statistics is hard to quantify because of all the missing data and everything in our data. So we decided to do a permutation approach, and we use a decorrelated residuals because our residuals are correlated that naive permutation doesn't work very well. But these are also like technical details, which I'm not going into too much. So I just want to say how the model fits the specific data. So here, the first analysis that we did was that, I hope you can see this. This is a little blurred in my screen. Generally, what we want to see is that the first model we test is that whether the first slide gives the model estimation with no interaction. So essentially, we have 7 different bacterial species and 6 plates. We see the beta estimate. Again, this is only one representation of all the possible betas that will give us the equal fit of the model. But what we see was that the plate 1 and plate 2 have a similar beta estimate. 3 and 4 have similar estimate, 5 and 6 have the similar estimate. And when we plot them in the principal component analysis, so essentially you can think about plates that are closer together are more similar with respect to their beta estimate. And we do see that plate 1 and plate 2 are similar, plate 3 and plate 4 are similar, plate 5 and 6 are similar. So we looked back. We initially did not know why this is the case, and we looked back to the original data and asked the persons who generated this data set. It turned out that plates 1 and 2 were processed in the same way. Again, remember we have the cell samples, the DNA samples, and the PCR product samples, which our estimate clusters naturally with different plates. And when we do a hypothesis testing, each of the plates have a different bias factor with the rest of the plates, except I think only plate 5 and 6 are not differentially are not different with respect to their bias profile. The second test that we do was that we try to test for the interactions. So the first model that we do is that we apply our interaction model to all the data. While adjusting for the plate effect, you see that here we gave the p-values from 10,000 permutations, that there's no significant interaction effects here. So this is this actually supports what MWC model initially proposed, that the interaction effect probably is not as significant, as important as the May effect model. When we look at the specific plates 1 and 2, 3 and 4, 5 and 6, which are the plates of the same type, we sometimes do see significant differences in plates 1 and 2, which are the places where the May effects are the smallest. The May effect difference are the smallest. Where the plates 3 and 4, 5 and 6, there's generally no significant difference with respect to their bias profiles. We also do hypothesis testing, and we found was that the interaction effect, even when it was present, such as in plates 1 and 2, the interaction effect was much smaller in the magnitude compared to the May effect model. And we were quite happy to see this result, because this essentially says that we may be able to characterize the complex bias generation process using something that is simple. So finally, use the final 2 minutes as a discussion. Currently, this model only works for mock communities when we have known relative abundances, because we need to know the differences between what you observed and the true relative abundances. Some ongoing work is carried on in our lab, in which we try to extend this model to situations in which we do not know the true relative abundances. Of course, that will require additional assumptions on the model. Statistically, we propose a multivariate permutation with when we have structure missing, and this can be have general applicability to other statistical contexts. What we think is the most important was that the fact that there's very little interaction, or there's no interactions between the biases between taxon and taxon, opens the doors to many future analyses. For example, we may be able to design models that are bias resistant because of the lack of the interactions in the bias generation process. And finally, I want to thank my collaborator Glenn Stanton in doing a lot of this work, and we worked together on this project, and he's the person who obtained the Brooks data. We also want to thank Michael McLaren and Benjamin Callahan, who are the major authors on the MWC paper, for nice discussions and critiques for our paper. And finally, we want to thank Paul Brooks for providing the data, and I want to thank our funding sources from NIH as well.